And let's go ahead and do this, should we? Alrighty. Well, welcome, folks, to our first virtual honors colloquium hosted by the Texas A&M University-Texarkana Academic Honors Program. I am your virtual host, Dr. Craig Nicashin, Director of the Academic Honors Program. And for those of you who aren't familiar with our honors colloquium series, every couple of weeks during the semester, we try to do a talk, usually by one of our faculty members or our honors students, about projects or research that they're working on. Dr. Ben Neuman is a regular in our series. He gives our annual holiday lecture on viruses. So we thought we would, drag him back out, and he was very, very happy, of course, to help us out here in April to talk about COVID-19, which has essentially, shut the world down, more or less. So the way this is going to work is we're going to go ahead. He's got a presentation that he's going to go ahead and run through. Once he's done, if you want to ask him any questions, go ahead and type them into the chat box. They'll come right to me. I will kind of ask them of him. And we also have Dr. Matt Young with us and Eric Schlaughter, who's the Emergency Management Coordinator for the City of Texarkana. So if you have any questions specifically for Texarkana sort of implications, let me know and I can pass those on to them. Or if Ben wants to punt them over to them as well, that's fine too. Yeah, there we are. So I am recording this. We will post it on the Honors Program website, which is just TAMIT Honors Program on Facebook. And then I'll try to make a link to that available for anyone that has missed it. So, okay. I will go ahead and mute myself and probably turn my video off so you can focus on Ben. And yeah, take it away, Neuman.
All right, let's go. Let me show you yourself for a minute. Yep. Sure. And let me show you this for a minute. Oops. Oops. And there we go. So what are you seeing? Do you see just the picture?
Yeah. Well, we see just the picture, and then there's a little box of you on the side. So SARS, COVID-2, and you.
I'm dumping me down in the corner. There we go. That's a little better. Whoa.
There you go.
Back here. Okay. All right. So there we go. You know what? If it shows up, it shows up. If not, we didn't need it. Hi. SARS-CoV-2 and you. This is the talk today. So what I'm going to go through is some of the latest information. Some of it only came out of embargo literally minutes ago. And I'll show you some of the things that we know already from the last SARS outbreak, because this new virus is very much the same as the old virus. It's a direct relative there. Two apples off the same tree. So let's go. There we go. Okay, so first of all, there are currently 7 known human coronaviruses. These are coronaviruses that can infect humans. We think 4 of these are human native. So those are going to be the 4 down here at the bottom. And they have all these kind of sci-fi names like HKU1 for Hong Kong University, NL63 for Netherlands. I think it was Sample 63 or maybe Patient 63. And OC43 and 229E are just, their names are shrouded in mystery. Yeah. Then you've got a couple other interesting ones. So these guys are usually seasonal common colds, and they're fairly mild, but they are all relatives of the bad ones. So the group we're in are called beta coronaviruses. And so that's everything from here down to here on our tree. Shown over here, these are the alpha coronaviruses, and we also have gamma, delta, and epsilon, of course, because everybody likes those Greek letters. Up here at the top, you can see some of the bad ones. They are outlined in red. These are the serious coronaviruses. And so you have original SARS coronavirus, which came out in 2002 and got really big in 2003. And then came back again a little bit in 2004, thanks to we weren't sure if it was a laboratory incident or a second infection, something like that. We've got the Middle East Respiratory Syndrome coronavirus down here. This is a thing that's been coming back every year since I think around 2012 or 2013. And then we've got the SARS coronavirus 2. That is the new virus, the one that causes COVID-19. And so we'll get into the naming in just a minute, but that gives you an overview of roughly where we are and how things are related. So I was on the committee that came up with the name. I'm not in charge of the committee. That's a guy called John C. Burr. So if you don't like the name, he's the guy to go see. But this is a little overview of where the name comes from. So let's start down here at the bottom first. So originally, back in 2003, the committee, and this is before I was on the committee, came up with the name SARS coronavirus, but they got it from the name of the disease, which was severe acute respiratory syndrome. All of these coronaviruses that we're talking about, MERS, SARS coronavirus, and SARS coronavirus 2, could be said to cause some version of severe acute respiratory syndrome. But since the name was already taken with the first one, we have to come up with other increasingly weird names to call the later ones. So the World Health Organization are the group that actually gets to decide what the disease is called. And I'm on the group that's the International Committee on the Taxonomy of Viruses Coronavirus Study Group. Yeah, that's a mouthful. And their only function is to put the viruses, attach them to their family tree, and come up with standardized names for them that everybody can then use. And so the disease is what informs the name of SARS coronavirus. It's the coronavirus that causes SARS. From there, we found this new coronavirus in 2012. And so it was from the Middle East, but it caused basically SARS-like disease. So it was called the Middle East Respiratory Syndrome coronavirus. And the WHO reluctantly named the disease Middle East Respiratory Syndrome after the virus. And then, I think this is important, really regretted it because Middle East, we're name checking a particular part of the world. And it makes it seem as if that part of the world has, bad things in it, which every single part of the world has bad things in it and different viruses. Yeah, rest assured. And so they're trying to now not name check a place or a person or anything concrete other than a disease with a name. And so they'd rather have a made up nonsense word than, for example, Ben Neuman virus, which I guess is fine. Let's see. So over here, when we got the new virus, the committee looked at this thing, and genetically, the virus is very similar and actually groups right down there with SARS coronavirus. And it groups within a bunch of viruses that are all called SARS coronavirus, but only 2 of these have ever been found in people. So there are lots of kind of SARS coronavirus, and they're all viruses of bats. And I'll show you some adorable pictures of those bats in just a minute. So we named the virus SARS coronavirus 2. The World Health Organization wanted to wait to actually name the disease. They did not like this because their job, and this has been put out in print now so we can talk about it, their job is to communicate with the governments of lots of different places. And I think the pushback they were getting was from some governments, perhaps China included, who were saying that if you call this thing SARS, it's going to panic people and it's going to make it difficult for us to actually stop it. And so they are very careful to always go out of their way to never mention those 4 letters in that particular order, SARS. What they'll say is that it's the COVID-19 virus, the virus that causes COVID-19. None of these are official names, but that's why they're doing what they're doing. And I support that. They're doing an important thing, and that's okay. So they named the disease, it's COVID-19, which is coronavirus disease from 2019, which is okay. But of course, remember, there were 7 different kinds of coronavirus disease circulating in, actually 6 in 2019. Original SARS was not. And so, yeah, I guess that works for a little while. So where do these viruses come from? The answer is there are new viruses, new to us anyway, out there in pretty much every kind of plant, animal, and bacteria on the face of the earth. So over the last 2 years, we've discovered corona-like viruses in all of these organisms. So you've got Eurythemera, which swims in the ocean, a little copepod. You've got Aplysia, which is a giant sort of sea slug that's really neat. Microhyla is a little frog. We've got an axolotl coronavirus that honestly I should be talking about at the coronavirus meeting right now if that thing wasn't canceled as well. And a bunch of other ones, other really neat viruses. And so we've got a lot. Originally, we only knew about coronaviruses in animals, but we've since figured out, rather in like barnyard animals and people, we've since figured out that the wider group of viruses is present in pretty much every kind of animal on the planet, ocean, freshwater, or land. And so that's pretty neat, actually. And so each of these viruses has the same basic equipment as the new SARS coronavirus, but right now these viruses do not infect. So we'll go through maybe one more slide, and I'm going to take you through the life cycle of the virus, or the replication cycle, because it's not really technically alive. And we'll go part by part and show you what each of the parts does and some of the work that's been done on them, because there's been a ton of work really quickly. I think we're going to start with the disease, though. So first of all, where does this come from? The answer is we don't know for sure right now. So shown at the top is the Malayan pangolin, or some people call it the Java pangolin, or it has a proper scientific name as well. There are lots of viruses similar to the new virus that have been found in pangolins. There was another batch of those published last week. We also have sequences from a bat. So this little goofball with the buck teeth down here is Rhinolophus affinis. And this is a teeny weeny bat. It's about the size of a chicken nugget. And they are found all over, all over Asia. So China plus, greater Asia. And so you can see that's the tip and fingerprint of somebody's finger on there. So you can get a sense of the scale of these things. They're just microscopic, not quite microscopic, but really small anyway, little bats. And so pangolins are, one, adorable, and two, they're a sister group to all the other carnivores, things like cats and dogs and bears and seals. So that's pretty neat. So that's the new virus. That's the COVID-19 causing SARS coronavirus 2. The original SARS coronavirus also came from the same kind of bats. So this is Rhinolophus affinis, and then meet Rhinolophus ferrum equinum. And this is one of 3 bats that have been found to carry the SARS coronavirus, the original SARS coronavirus, or viruses that are very similar. It seems that these bats carry the virus for life, and for them, the virus is going to cause a little bit of diarrhea, or rather it's going to be enteric. So it'll come out anywhere that they defecate. And so this is thought to be the start of these infections. Humans do not normally eat bat guano as part of their diet, but there are a lot of kinds of animals that would snuffle along the ground, and these bats are just flying around in the world. And so we think that is probably how it gets picked up. Some other animal gets contact with the virus through that means and then passes it on. So with the original SARS coronavirus, it came to us from the bat, but via these little guys, which are called a Himalayan palm civet. And they actually farm them for meat over in China. You see them in zoos over here sometimes. They're also a pretty adorable animal. And I don't know, I guess delicious, maybe. The palm civets got the virus, and they actually suffered from it far more than humans. It was something like 98% fatal in palm civets, if I'm remembering the stats correctly, and only about 10% fatal in humans. So we know, though, that the SARS coronavirus is part of a larger group of viruses that are also called SARS coronavirus or SARS-related coronaviruses. And so the difference is we're going to have SARS coronavirus is the original one that infected humans. We're going to have, this paper came out early in the infection before the name was public, so they're still calling it 2019 novel coronavirus. But that's going to be your SARS coronavirus 2 up here. And everything down here in red and gray and blue is some form of SARS coronavirus. So genetically, these are all SARS coronavirus, but there are some differences. So there was a group that was just about to publish this paper when the new coronavirus hit at the end of last year, beginning of this year. And when that happened, they were able to throw in one little test with that one, which is shown up here. It's panel D, clearly tacked on at the end. And so what they were looking at here was, can the proteins on the outside of these viruses actually grab onto and infect human cells? And so they did it on different kinds of human cells that had different, what we call receptor proteins. The receptor protein is something that is on the outside of your cell normally, that's part of what your cell needs to do in order to stay alive. And the viruses are they are adapted to grab onto the outside of these, and that's how they get entry into our cells. They latch onto a protein on the outside. So the original SARS bound to angiotensin-converting enzyme number 2, which is part of a system called the angiotensin-renin system, which is one way that we regulate blood pressure in the body. So angiotensin-converting enzyme 2 will convert one form of angiotensin to another form of angiotensin, giving it an effect, basically. And so what you can see up here is that, let's start with panel D first. Angiotensin-converting enzyme 2 is a good receptor for the one that infects people and the original one, but also for these guys like SHC14 and WIV, and probably all the ones in red down here. This is the same test with more viruses. So all the rest of these guys with these made-up looking science-y names have not yet been found in humans. And so we don't think they are able to infect humans, but these are genetically very close relatives of original SARS coronavirus and SARS coronavirus 2. But I guess the other interesting thing is here, look at all these gray ones down here. These guys do not infect via the SARS receptor or via the MERS coronavirus receptor here, or via the other common human coronavirus receptor. This is the one for coronavirus 229E and NL63. And they don't infect cells where you don't deliberately express any of those receptors either. And so, yeah, really, we think that these may be close, but probably don't have the right combination of proteins to be able to bind onto a human cell. So what a virus does is it's essentially going to do everything for itself, but it needs all of the access to all of the equipment that's found inside of a cell. And so that's why viruses have to get into cells, and it's really the step where the virus gets into the cell that is the main determinant of whether or not a virus will grow. Most viruses will grow in different cell types if you can get them in, but yeah, usually the blockade is at the level of entry. So yeah, if it can't get through the door, you don't have to worry about it for the most part. All right, so what does SARS look like in the clinic? So here are some radiograms and a little section from a CT showing you patients with the original SARS, and then the little CT that I pasted over the top, that is the COVID-19. And so what you're seeing here is, so this is the lungs, and you got the heart here, and the lungs are going to stop right down there. The area in here should look just black and totally blank like this. And if you start to see any of this stringy stuff, like down here, this sort of white cloudy stuff, that is a mixture of fluid and scar tissue and white blood cells that are infiltrating the lung. And on the one hand, that's okay because they are going to clean out the virus. But on the other hand, they destroy the part of the lung where they are attacking completely, as in they break down the alveoli so you can't get any oxygen from that part of the lung. And so it's going to proceed. It's going to usually start out and happen in the middle to lower half of the lungs. The virus is very much a lower respiratory virus. If you had something upper respiratory, it would be causing problems up in here. And you can see that's pretty clear. This is a very severe patient. I believe from the paper that this person actually died of the original SARS. And you can kind of see why. Even with a ventilator on, look, you're missing one lung and about half of this lung. There's just not enough space there to be able to oxygenate the blood. Unless there is some other machine on hand that can do that for you, there would not be a lot that modern medicine could do for this person. And these machines do exist, but there's a limited number of them. And yeah, it's first come, first serve. Doctors try and do their best, as do hospitals. So here's a little time course. This is at the start of symptoms, so probably about a week after infection. You can see it's happening in one side of the lung only. The other side still is clear. By the second week, and there are little dates on here, on the 21st, you've got it filling up both sides of the lung, but still only the bottom part of that, because that's where the receptors for the virus tend to hang out. And then you go a little further into the future, and you can see that this person actually starts to get over it at this stage. And so we're a week later, they're over their symptoms, but see all that scar tissue that's still there? And then this is even a week after that. There's still a considerable amount of scar tissue in there. So from what we know so far, the shortness of breath and the damage to the lungs goes on at least for years. It's only been 18 years since the original SARS outbreak, 17 years for most people that caught it. And so we don't know quite how long this can go on for, but the last thing that I read, which was about 2 years ago, was talking about people who were still having some problems breathing. And that's going to be correlated to how severe the disease was at the time. People who are younger are going to be able to repair that damage faster and probably will have less damage overall. And so they tend to fare better. People who are older, their stem cells just aren't as good at repairing damage. And so, yeah, they would probably have more long-lasting effects. Shown over here, all these bits that are discolored, that are a little bit whiter than they should be, those are the areas of damage. And so you can see this is right around the middle of the lung, and you can just see, look at all that damage caused by the SARS coronavirus 2. So it's very much the same thing, just a different way of visualizing it. Okay, this is the hot new paper. This one is coming out, or says it's coming out of embargo today. I'm assuming those usually happen at 10 a.m. East Coast time, occasionally 11 a.m. But either way, we're okay. So hot off the press. This is from Germany, from another guy, let's see, Christian Drosten down here is also on the committee that names the coronaviruses. And he is a medical doctor. There are three medical doctors that I know of on that. Actually, no, 4 on that committee. And the rest are science doctors like myself. So what they are showing here is they're trying to answer the question of how do people get infected? Where does the virus grow? How much virus is there? When is the virus there? When do you start to be able to detect the virus? And they do this all in 7 or 8 little panels right here. It's really wonderful. So shown here, we have people that are reporting to the hospital on the day when they first have the symptoms. Normally, the test that's done is they're going to put a swab down either in the nose, or it's better to put it in the throat and try and collect a little bit of virus from there. And then if they happen to get enough virus, then they can detect that thing. It turns out from this paper that they have to get about, here we go, 10 to the 6th copies of RNA, which is about 1 million copies of RNA on that swab. So 1 million individual viruses is the point at which you can first detect a positive culture. So people who have early stage infections or low levels, the virus is not going to be detectable. But if it's not detectable, it also can't infect other people. So this is a little bit of good news, I would say. The people who are reporting into hospital on day 0, only 1 out of the 9 patients in this study had any detectable virus, and that was found in the sputum and via a swab. So this is virus that's growing apparently in the throat. And they confirm in this paper that the virus does actually replicate in some of the cells in the lining of the throat, in addition to replicating down in the lung. And then you can see that over time, you start to get the virus in other places. Interesting thing here is that you have virus that's present in stool samples, actually in very high amounts. And the other interesting thing here is that, so the person's going to feel sick at this point. By about day 7, they're feeling very sick and would be in the most danger. But by about day 14, the person is going to feel better and is going to want to go home, or will be released in a lot of places. But you can see here, there's still virus that's detectable in the sputum, so it's still right there in the throat, and coming out in the stool samples, even for, it looks like about 2 weeks after the person would be cleared of the virus, and after they would no longer be able to detect the virus in the throat. So what they're doing here is a different kind of test. It's a test that virologists do, where they can actually count active viruses by using them to infect cells. It's a time-consuming method. It's not something that most hospitals would want to do because you can't really scale it up. So the test, which is a PCR-based test, is really much better for doing that sort of thing, testing large numbers of people. And the other thing people are asking about is antibody tests, and so that's kind of hiding up here. Let's see if we can move this up. Okay. So here's the antibody tests, and this is showing that these people do not have antibodies until the first time you can detect them in a little under half the patients. was about day 5, and then they don't all become positive until about day 9 or day 10, which is when you're getting close to releasing them from the hospital. So serological tests are not going to be particularly useful in diagnosing acute infections, like while the person is still infected, but they will be massively useful because we think those antibodies are going to stick around for at least 2 years. This is going to be a great epidemiology tool, so we can go back in after there's been an outbreak and test people around that person and figure out who had it, where it came from, how big was the problem, and where it might flare up next. So that's how those tests are really going to be used. And there are a couple of those that are commercially available. They are shipping either at the end of last week or the beginning of this week. All right. Here's the data from the first Chinese CDC paper, and it's held up pretty well through subsequent studies. The overall case fatality rate for SARS, this is people that we know are infected, and this is for Sars Coronavirus 2, COVID-19 disease, is actually only about 1.4%. There was a nice paper that recently calculated it at about 1.38%. And it is not evenly distributed across all people. So the virus tends to infect people who are in sort of middle age or older, which looks weird. You would say, what are middle-aged people doing that kids aren't doing? And the answer is nothing. And kids live with those middle-aged people in many cases, or visit them on the weekends at grandma and grandpa's house. So what you're seeing here is that younger immune systems tend to be able to deal with this virus. I think the exposure is the same across most of those age groups, but younger people tend to be able to deal with the virus so quickly and so effectively that it doesn't ever show up as a positive. Because if you never get to a point where you have 1 million viruses on one swab in your throat, then you are not going to have a detectable case of COVID-19. And so they'll think, oh, this person's sick or may have had contact with somebody, or this person is fine, but they won't know for sure. You won't be counted in the statistics. So these would be the completely asymptomatic people. There are probably, there are probably more people, particularly in the young end, who are being exposed than are being counted. All right, so here's from back in the day. This is me and I think 04, and that's my lab assistant, and we needed to trim her little hood. The hoods were really long. They're supposed to come with a second suit that goes around the outside for double protection. We were conserving suits, and so we were ethanoling and cleaning off the outside of the headgear. So this is hooked up to a little respirator, which is like a little tiny backpack that you wear on a belt. This is a thing called a PAPR, and it's got a couple of HEPA filters in there, just like you have in your vacuum cleaner. And so they are filtering the air, and then it's running up a tube on the back of the gown there and into this. And so all you're doing is pumping air over your face and then down outside of the hood. And because the air is flowing in that direction, it means that the virus is not going to flow back up inside your hood. And as long as you don't do anything colossally stupid, like reach your gloved hand up inside of there to scratch your face, which, yeah, we did not do, then really there's no way to get infected. And it's a nice, safe way to work with this. You actually don't need this level of protection to be able to be safe from the virus. What we were doing is concentrating very, very large amounts of the virus, like concentrating 100 billion active virus particles in a little tiny pellet at the same time. And we were doing this so that we could take pictures like this. This is a cryo-electron micrograph of the original SARS coronavirus. And since the new coronavirus is also SARS, this is exactly what the new coronavirus is going to look like. We're hoping to work with a group out in California who are going to get access to the virus and have the facilities to take pictures of the new one, and then we can check it out. And so over here, this cool sort of purple tube thing shows you how we purify the virus. You make a gradient from very thick to very thin sucrose, so just sugar water. And the stuff at the bottom is almost like honey. And a virus is just a little mechanical thing, and they will all band at a particular density, whatever the density of the virus is. You just put them in a centrifuge and run them for an hour or two. And these we actually stained up with a little green dye that shows you where the virus is. So it's this entire layer right here. And it's going to be sitting at about, normally at about 38% sucrose. And that's weight per volume. No, weight per weight, sorry. Okay, so new research. This came out extremely quickly, actually toward the end of February, and it's some Texas science here. This was done by a couple of guys over at UT. And so they're also using cryo-electron microscopy. It's a giant instrument that costs something like $1 million, and plus is very expensive to run. So probably not something that we could do very easily at A&M Texarkana. But they got the structure of the new coronavirus spike protein. And so there it is. That's what it looks like. And the really neat thing about this, so you can see all the domains. And if there's anybody out there who's in my virology class, these are the two heptad repeats down here. They're going to zip together and they're going to fold the second half of this basically in half. So this little blue box right up here is called the fusion peptide. That is going to act like a fish hook. So that's going to sink into the cell. And the area down here is actually buried in the virus. So that's where it goes through the virus membrane. And so we hook the cell like you'd hook a fish, and then we crash this HR1 into this HR2 down here. And that reels in the fish. So it crashes the virus into the cell, and that's how the infection is going to start. The neat little thing about this structure is that it shows for the first time that there are actually two ways that the spike looks. So there's 3 copies of this spike protein, and this is the one that makes a coronavirus a coronavirus. It's the one that sticks out on the outside. There's going to be 2 copies that are in what we call the down conformation, like so, with the green thing pushed up against the rest of the protein. And 1 copy is going to reach up, and this is like a little roving hand that can kind of have a little extra reach out and see if it can grab onto a cell. So think of this as like one of those little grabber claws that you can use if it's hard to pick up things off the floor. And the virus has one built in. It's a very efficient protein in terms of getting the virus into cells. Here is from another piece of work. This is from one of the 2 papers that came out last week, and these were both talking about the structure of the spike and the receptor and where they come together. And so the blue thing down here, this is the receptor, and you see it has this nice curved helix at the top. This is the edge of the spike proteins. This is that little bit that was either in the up or down conformation, but just a little green bit at the top, not the rest of the protein. And as you can see, it just sort of cushions right around that nice little thing. There are little molecular interactions shown here with the little red lines, and this is how it's holding on. And you can see on either side down here, we're kind of wrapping around the end of this, it's called an alpha helix, this little twisty cord. And so, yeah, a nice sort of curving structure in the center and a little clamp on the end, and that's how the virus is able to bind to the receptor. Now, we don't know what the receptor of this particular bat looks like, and we don't know what the receptor of the pangolin looks like. But we do know that all mammals are pretty closely related, and so it's almost certainly only a slight variation on what you see here. And probably it just happens that this alpha-1 alpha helix is similar enough between pangolin, bat, and human that it looks like the virus doesn't actually have to adapt at all. We have not yet seen any mutations in the part of the virus, the yellow thing up here, that comes into contact with the receptor. And if there was an adjustment period, like we saw with the original SARS, you would expect there to be some changes that occurred in that area. Okay, and let me show you a little bit of experimental stuff that we did. So the spike has these fusion peptides, and this is how you can identify one of those. So what we've got here are little green fluorescent vesicles. It's like a little soap bubble, basically, made out of membrane lipid. So I want you to watch this guy up here. So let's rewind this, go back to the beginning. See, it starts out nice and round, and then look, it just gets all of its sides pounded in. So what we're doing is we're putting in the little thing that we call the fusion peptide. We made a little tiny protein, put it on there, and you can just see it embedding itself in the outside of these vesicles. And if you give it long enough, it'll basically turn them inside out. You'll just have all this protein crowding for space, trying to get onto the membrane of the vesicles. And yeah, there you go. You go from sphere to hexagon to kind of a wonky pentagon. And if you let it run all the way to the end, yeah, it just kind of folds up and sort of crumples in on itself. Here are some still images from this. And so we can show that this happens with the fusion peptide. So here's the MERS coronavirus fusion peptide. And so we go from this to just smaller and just there, completely crumpled over 5 minutes. Here's the mouse hepatitis coronavirus. It's a relative fusion peptide. We also did this for the SARS coronavirus fusion peptide, does the same thing. Other parts of the spike do not do anything. So here are two other parts of the MERS spike, and here's a part of the MHV spike. And the vesicles, they wobble, but they don't really do anything. And over here's a positive control. So the M2 protein of influenza virus is also known for doing this. And you can see that's what was considered really strong protein insertion into a membrane up to this point. And just look how ridiculously much stronger the coronavirus fusion proteins are compared to that M2 protein. So yeah, it doesn't make the virus any more dangerous. It just shows us a little bit of how it works. Okay, so once the virus gets into the cells, we've done the spike receptor binding, and the fusion peptide inserting into the membrane, and then we think it crashes the virus into the cell and dumps the virus contents inside there. The contents of the virus are one long piece of RNA, and that long piece of RNA encodes about 30 proteins. And the 30 proteins make a little thing like this. So this is a replication factory from the chicken coronavirus. This is a paper I helped out with a couple of years ago. And look at that. So each of these little chambers is lined with proteins, and each of those proteins is going to be making new copies of the virus. So this is what the virus actually looks like when it's growing and kind of alive inside of a cell. This is a 3D electron microscope image, so everything you're looking at is very tiny. And we're scanning from the top of the image down to the bottom of the image, so that's why it's changing. We're just looking through three-dimensional slices of the same thing. But yeah, that's what a virus really looks like. Gaze upon its face. It's pretty neat. So after we have made those little replication factories, and we did a paper a few years ago where we figured out which proteins actually are responsible for making those factories, and it turns out there's 3 of them, but only 2 of them are really important. One of them seems to hop along and sort of gang up with the other 2, but it doesn't really do much on its own. Then the virus polymerase is going to start copying viruses, because that's all the virus does. It makes more copies of itself, and it sort of shuts down the host immune system so that it can make more copies of itself in peace. Yeah, like you telling your kids to, be quiet. Mommy or daddy, needs to sit and think for a while. So we got the structure of that protein. This is from the cat coronavirus, but it'll be almost identical to what you would get from the SARS coronavirus or the SARS coronavirus 2. There are 3 different proteins that come together. Protein 7, NSP7, protein 8, there are 2 copies here, and protein 12 is the polymerase, and it has 2 domains, Neuman and the polymerase domain. And that little interface thing is just like a brick that holds everything together. So we now know what the machine looks like that's actually going to copy the RNA, and that's another great improvement. I've studied coronaviruses for 24 years, and all that time I was saying, why do all the other virus families get to know what their polymerase looks like and get all the cool things? Finally, we're starting to get them. Finally, the understanding of coronaviruses is catching up. Why do we get coronavirus disease? Well, here's why. Because the virus is really good at the game of hide and seek. So normally there's a system inside your cell called the MDA5 system or the RIG-I system. So it's these 2 things up here. And what it is a protein that'll open up like a little, like a switchblade, basically. Yeah, boom. So there's a little blade up in the air. And once you get 4 of these things opened up together, you'll attach another protein called ubiquitin to the outside of those. And once you got 4 of those with ubiquitin all grouped together, they will signal to another one that's called the mitochondrial antiviral signaling protein, or MAVS. Yeah, not named at UT Arlington, who I think are also the MAVs. Then we've got, it's going to go on to make some factors that'll go into the nucleus and make an immune response and block the virus. So the virus carries 3 proteins that specifically prevent this protein from recognizing the end. So what it's looking for is ineffective capping, and that's how it knows it's looking at a virus RNA as opposed to a cellular RNA. And then you've got another protein that comes in, and when we try to attach these little blue tags to the top, this protein, NSP3, will actually cut these little tags back off again. And so we've silenced it. MERS coronavirus packs an extra protein called protein 4 that is going to block the signaling of this protein, of the MAVs. riding along like a little cowboy on the outside of the mitochondria in that weird drawing. And then if we do happen to go to the nucleus and start to make messenger RNA, before it can get translated, a protein called NSP1 will physically destroy all of the messenger RNA that the cell is currently making. And there's another protein called NSP3. It's the same one as up here, but it has a different domain, a different part of it, that will block the cells from storing up copies of interferon RNAs in a thing called a stress granule or a P body. And this just goes in and assassinates both of those. So the virus is doing a very good job of covering its tracks, and it's kind of a race between the virus and the host. Can you sound the alarm before the virus cuts all the wires and you are unable to sound the alarm? That's the dynamic that determines how well it goes in SARS coronavirus or COVID-19 infection. So here are some pictures of the actual virus. So these are some cryo-EM shots that we took of some coronaviruses. So let's see, we've got the mouse coronavirus, we've got the cat coronavirus right there. We've got the original SARS coronavirus looking great. We've got the mouse coronavirus in a slice from a three-dimensional image of those things, again with an electron microscope. And then we even went back and tried to figure out, well, what does it take to build a virus? And so we put together some of the proteins. So here's the M and E protein. And sure enough, you can make little things that look pretty much like viruses. And if you put the N protein in, then you get a little better organized version of the same thing. So let's see, we've studied the way these things come together. So when the virus is getting ready to leave, what it has to do is it has a protein called the M protein, which stands for the matrix protein, which is a pretty cool name. This thing starts out flat along the membrane, and we call it the M compact form. And then when the rest of the components come together, then it will convert. It's like a little transformer, and it'll turn from the M compact form into the M long form right here. The M compact form is sort of flat in the membrane. So you get that wherever the membrane is flat, like up here, and this is an interpretation of that. The M long form is going to curve the membrane everywhere that it is, or it's going to try to do that. And so here's some nice refined images of the M proteins all pushed up against each other. And those are still the best pictures we have for the M proteins of any coronavirus. The other big protein on the inside of the virus is going to be the nucleoprotein. This is like a little chaperone or a personal guard for the virus genome, for that virus RNA, because that's really the instructions that make new viruses. That's really what the virus is. All the rest of that shell is just a delivery vehicle and a protection detail. It's really all about the genome. And so this is showing a model for what we think the genome does when it binds to N. So we think there's a little thing right up here, and so it looks like the N is face-palming itself basically with its domain N3. You can see it right there. And when a part of the RNA called the packaging signal comes along, which is only found in the longest form of the coronavirus RNA, it's going to get in there, bind to the same spot, and it's going to pop off the N3 region, which can then reach out and interact with the M protein. And so once it does that, it pulls the M protein down into that long conformation, and yeah, that's how we get budding happening. And the membrane is going to bend the other way once this happens. Yeah, not going to bud into the cell. That would be silly. So another interesting thing, each of the proteins that is incorporated into the virion, that's the virus particle that looks like this, each of those guys actually plays a role in helping the M protein be the best M protein it can possibly be. And so this is just because it's kind of funny to be able to say. The M protein works most efficiently when it has all the other proteins there. So you get some viruses out that just were born without any spikes on. So we called these bald virus particles. And it turns out that the bald virus particles, so the less spike they have on them on the outside, the larger the particles tend to be. So baldness and obesity are correlated in coronaviruses, which is very unfortunate for them, but it's okay. We can make fun of them. Yeah, they're a bunch of little jerks. All right, and so here's the model for how all this comes together. And so this is actually the thing that would infect you, this little biological machine. You've got your RNA squiggling around the inside, that's the little lines. You've got your nucleoprotein, the N protein, kind of wrapped around it and protecting it. You've got your M proteins in the long conformation and short conformation, interacting with the spike proteins and a couple other minor proteins shown there in yellow. And this just shows you how it all comes together. And this is the graph that shows you what I mean when I say that it gets a little more efficient, the entire process, when you get all the parts together. So if you start out with just vesicles, they can basically be any size. So they're this sort of light blue, just smear over here. They're all over the place. And so that's just a membrane with no proteins in it in particular. When we add the two viral proteins, E and M, which are the minimum set that you need to make new virus, then you notice it tightens up that distribution. And every time you add another protein, so here we're adding the N protein, here we're adding the genome, and here we're adding the spike on the outside of it, we get to a narrower, better formed version. It's actually going to use up fewer proteins and be a little more efficient. So you see the standard deviation, like the variance, goes down. This is just a mechanical manufacturing process. And the size also goes down, so it gets a little more efficient, the more of these proteins you put in there. So that's the role of each of these proteins. The M protein is bad on purpose. It's bad at leaving on its own because it doesn't want to leave before it gets everything else together. Because if the M protein shoots off too early without any spike proteins, then there's no infection at the next level. It just wasted a bunch of M proteins. All right. So inside the coronavirus, we know there are a lot of things. So this is from a paper we did a few years ago looking at the Sars coronavirus. These are all human host proteins that you find on the inside of Sars coronavirus. So you've got all kinds of things in there. It's basically a scoop of the inside of the cell. We think there may be a couple of proteins on here that could be important to the way the virus causes disease. This is known for some things like HIV, for example. But honestly, it's a giant list. And for most of these, we don't really have a good idea of whether or not this protein is important or if it's just part of the junk that the virus picks up. One that we know is important is FASN, the fatty acid synthase. We know that if you slow this down even a little bit, the virus really suffers and just cannot grow. It can't make that little factory for itself. And the virus just withers and dies, which is really cool. So these are some of the people that have helped me over the years. I'm probably missing a couple of people off here, but I think that's a good point to take some questions if there are any. So yeah.
All righty.
How do you want to run this? Yeah.
Thank you very much, Ben. I know we had a couple that came in. So once again, for folks who are kind of joining us, if you want to ask a question of Dr. Ben, go ahead and just put it into the chat box, and that will come to me. It occurs to me, I guess I probably should have introduced you. I just kind of assumed everyone knew who you were. You're the chair of the biology department here at Texas A&M University-Texarkana, and now certainly world-renowned. Reached 1 billion people, you tell me, and on viruses. Fantastic. So all right, let's see. I know I had a couple of questions that came in already. There was one for Dr. Young as well. So Amy was asking, and I don't know who you want to take this question, on the suggestion of wearing a mask for healthy people to not spread the germs. Why has that not been more widespread? The idea of having basically everybody wear masks all the time.
So I can give you the common explanation that's out there that I believe, and if you want the actual medical explanation, you can go to the pros. The idea is that if you wear one of those little sort of cheap cloth masks, not the N95 that's fitted, what that's going to do is it's going to cover your nose and mouth. It's not going to prevent anything from necessarily going in there, but it's going to stop you from touching your nose and mouth. And so it's a nice little memory aid, basically, to help make sure that you don't accidentally infect yourself. The N95 masks are in very short supply. There are a couple companies gearing up to produce more, but there are big shortages of that, not just in the US, but around the world. And so we want those to be available to people who are deliberately suiting up and going into the room with a coronavirus patient to try to save them. I want those guys to have everything that they need in order to do that.
Okay. Thank you very much. All right, from Dr. Jordan, who might be giving our next honors colloquium in a few weeks. We'll see. He says, do we know to what extent some people may be carriers but have no obvious symptoms? Do we have a sense of what percentage that would be?
We're learning this now from that study that was released today. It looks like people are infectious for longer than we thought, longer than we can detect the virus. But it looks like they're not particularly infectious before they get sick. So it does seem from that paper and from some of the other figures I didn't show that the sicker you are, the higher the level of virus in your body. So people like kids that have very mild disease are less likely to spread it to other people, if that's of any comfort.
Okay, thank you. All right, another question. If people shed the virus for 28 days after first symptoms, are current quarantine recommendations in line with that?
No, they are not. Well, actually, it would be about 2 weeks from the point at which you were discharged from hospital. So it would end up being just about right, but only barely. Based on this paper, I would think you'd probably want to extend that to about 21 days after you were released from hospital with a confirmed case, or released from, releasing yourself from your bedroom with a confirmed case of SARS coronavirus 2.
Okay, thank you. All right, all right, so we have a question for Dr. Young as well. So if Dr. Young is still with us and wants to unmute himself and turn on his video, or if I might have to do that. Let's find out. The question is, essentially about the confusion around NSAIDs. Essentially what is, what is happening with those. So yeah, let me know if he can't unmute himself, and I should be able to do that. Oh, I have to unmute him. All right, thank you, David. Let me do that. While I'm looking for Dr. Young in my list of people, I did have one other question. What makes the differences in how long viruses can survive outside the bodies? So, what accounts for that?
Yeah, so there are viruses that they've managed to get out of the Siberian permafrost that are at minimum 30,000 years old. So if you put the thing in the freezer and pack it really nicely, yeah, sure, you can keep a virus around for a long time. But on the surfaces that we're talking about, like a surface in your home, there's nothing there that's 30,000 years old or is undisturbed for that amount of time. And so the virus breaks down pretty quickly. There's a nice paper if you were to go to the NEJM, Google NEJM and coronavirus, it'll take you to the page that has all their stuff. There's a study that came out very early in the outbreak that actually goes through every single surface.
I caused a problem there. Sorry about that. Go back, Dan.
Yeah, no, that's okay. Yeah.
Sorry, if you wanted to finish your answer, that was me getting in the way.
That's actually it. Yeah.
Okay.
Yeah, go read the paper and it'll answer whatever permutation of that question you have with actual data. Yeah. Yeah, the virus doesn't grow outside the body. It only breaks down, and it breaks down faster and slower depending on the case. It seems to be all gone within 3 days, no matter what the surface is, unless you're talking about something like a freezer where you're deliberately trying to preserve it. Yeah.
Okay. Another question, and essentially there's been a couple kind of about this, I suppose. Do you think if we had a nationwide shelter in place for 14 days, would that essentially end the spread?
Well, yes and no. Now we know that the virus is actually going to be around for about 28 days. And so we're talking about a month shelter in place. But yeah, if during that time you can find all the cases. So the risk would be if you had one person who just caught the virus going into quarantine, and then they slowly throughout the month infect the rest of the people in their house, then you've still got virus on the other side. So we need to really know where the virus is. That's part of why testing is so important, and also to model and figure out what this virus is doing. Get accurate intelligence on the enemy, basically.
Okay. Is COVID-19 heat resistant? If so, what's the temperature where the virus begins to degrade or die?
40 degrees, it starts to fall apart. Celsius, yeah. So like 100 and something Fahrenheit. The M protein, the one I told you about, just pops like a piece of popcorn. It is a very badly behaved, stupid protein. And that's the one that's going to take the virus down with it. Once a couple of those go, there's no way to get the genome back out of the virus. The RNA can't actually hurt you unless it is in an intact delivery vehicle with all the parts functioning at pretty near 100%. And so treatments like a little bit of soap or UV radiation that hurt one of those parts are enough to make it totally non-infectious.
Okay. All right, so going back to the question for Dr. Young, he says, with more research, we feel ibuprofen is just as safe as Tylenol. As we learn more, early research suggested possibly not. but we're beginning to feel that it's safe. So it's a bit of good news, I suppose. Hopefully that bears out, as this develops. Do you know where the efforts are with a vaccine? Like what has been, I know I've seen a few news articles, but
yeah, there are a lot of different ones. I know Johnson & Johnson aren't going to be starting their trial until September. The vaccine trials that are starting right now, there are at least 3 that I know of, are all versions of the same vaccine. So all they're doing is trying to either make that spike protein as a protein or as a messenger RNA or as a piece of DNA to make a messenger RNA to make the protein. And they put it into you in one of those 3 ways. It's the delivery that's different, not the vaccine. Candidly, I don't have a whole lot of hope that any of those will be a knockout. Coronaviruses have been very difficult to vaccinate against. People have tried experiments like this in animals before, and it's usually just not enough with just that one protein.
Okay. Let's see. Answering questions. Well,
but there's more on the way and lots of other things to try. Yeah, it's just they're going through the obvious ones first.
Sure. That makes sense. All right. From Dr. Casey, you'll like this. How does the aerosolization affect probabilities for asymptomatic transmission?
So it depends on how efficiently you think humans aerosolize virus. So we know now it's growing in the throat. And so theoretically, if you're breathing out or talking at somebody right in their face, there would be a small amount of virus that could potentially be coming out just as the air passes over that surface and comes right out of you. This is probably, so we've never had a really good mechanism for how any coronavirus infects. And as you can see with this virus, it goes into the lungs, just like that, causes that high hacking cough you just had. Thank you for demonstrating. But it also is going to go down the throat into the enteric system, and it's going to infect the intestine and cause diarrhea. And so I guess we're not sure exactly how that happens. It's hard to tell which one comes first, because on the paper that I showed you today, you can detect it in both places at about the same time. So it's really unknown, and it makes modeling that much more interesting and complicated for people like you, Dr. Casey, who are actually good at modeling. Yeah.
Okay. Very quick question, I think. Does washing your hands really make a difference in stopping the spread of the virus?
Yeah, washing your hands is going to pop that little bubble on the outside of the virus. So it's going to separate the spike protein from the M protein and the RNA on the inside. And once you've just pulled those components apart by a little tiny bit, there's no way to infect yourself without extreme patience and giant amounts of virus and a ton of expensive equipment. Yeah, you've got to really work hard at that point. So no, you're totally safe with just the tiniest amount of soap. It's, that's the wonder drug. Yeah.
Okay. What would your prediction be on a peak date range in the US and for Arkansas and Texas specifically?
I feel like in terms of internet memes and stuff, we have hit peak corona and then some. But in terms of when is this thing going to be over, that's a good question. So I was hoping to start seeing the numbers come down by this point, as you have most of the country, but still not all of it, in a shelter-in-place kind of situation. And it would be nice to get sort of an even distribution of these things across the entire country, because the virus doesn't care, and people who are not in shelter-in-place places can go in and catch the virus from people who are, in those places, and then it spreads. Yeah, the virus doesn't know about boundaries or anything like that. So I think right now what we're seeing is the rump of the original infections. We think there were probably tens of thousands more infections than the US knew about earlier on. And we are still eating into those, and we still probably haven't found all of those. So that's going to mess with the numbers. It's going to look like it keeps going up, but really we're getting through all the cases that were there all along and we didn't know about. And so that's going to make it a little harder to tell when it comes back down. And this is a case where if we're able to do more testing on just everybody who's even connected with somebody who is sick, then that is going to solve that. Now, it's not available in every place. Not everybody has the same amount of the test kits. And so in some places, they do have to kind of ration them and just give them to the people that need them the most. And so, that's a good question for our local public health people. Let them answer how they would like people to respond.
Okay. Just a note from Eric Schlauter, who's the Emergency Management Director for Texarkana, thanking you for your expertise and reiterating what you were saying about the masks and trying to save those for medical providers that are having constant and repeated exposure to this sort of thing. So I think that makes a lot of sense. A couple others.
So there's also, people have been looking at ways to sterilize those. There's no good way to sterilize those masks. So it's not like a doctor can put on the same mask and then go into a bunch of rooms. They can infect other people that way. These have to be disposable for them, which is why they need so many, which is why we got to not be using them ourselves. Yeah.
Okay. Do you know of any antibiotics that would be effective for this? I mean, I know, I don't usually.
Yeah, so there's a study that came out of France. I believe it had 6 people in the group that got azithromycin. So this is the Z-Pack plus an experimental, probably not great, but okay, anti-coronavirus thing called chloroquine. They used the hydroxychloroquine version. We've known since the 80s that this can work against coronaviruses, but it's never been all that good. And I really wonder if you're going to get enough of it to the lungs to actually be able to do much good. We have yet to see a big study come out, but as long as the study's going on, there's still hope. And yeah, maybe it'll work out. That'd be all right. But there are a lot of other drugs that people have tried over the 18 years we've known about SARS. And nearly all of those could be applied to the new coronavirus. It just takes a while and it takes a lot of money to try out each one. And so they're kind of going through them in order of who was first in line.
Okay, thank you. Let's see here. Another maybe quick one, I'm not sure. How effective, you mentioned how soap, obviously, washing your hands is the best thing. How effective is something like Germ-X?
99.9% effective, right? Just like it says on the outside. Yeah.
They can't print it if it's not true.
Yeah, sure. Would a bottle off the store shelf lie to you? No. Yeah. So the idea is that these can be effective, and they're doing the same thing that soap's doing. But the idea, as it was explained to me, is that so these things contain ethanol or rubbing alcohol, some version of alcohol. And so they're going to get in there and mix around with that membrane, that soap bubble on the outside of the virus. And then when the alcohol evaporates, it's going to punch holes in it. And then boom, at that point, it falls apart. We know that you can get virus back out of the ethanol before it evaporates, and it's still fine. And it's actually infected at least one person that I know of. So yeah, use the hand sanitizer, but then let it dry before you go, picking your nose or whatever. Yeah. Yeah.
A couple about sort of preparations that were made and maybe what the end game is. So I'll kind of combine these two questions. What do you think the U.S. could have done to be more prepared, and also recognizing that this is going to be with us, for a while? Essentially, what's the game plan going forward over the longer term, and how long do you expect this to last?
Okay, that's a lot. Yeah.
A couple of small questions for you.
Sure. Life, the universe, and everything. Yeah. All right. Let's go. Yeah, one at a time. Yeah. So I think what the response could look like is what the world response looked like in the Ebola outbreak, and I'd say that's pretty much the bar for taking things seriously. What we did there was we set up pop-up hospitals, basically a big tent, which we're now starting to see in some of the big cities. But pop-up hospitals and testing centers in the areas where you need them. We also saw major recruitment from the science communities and from healthcare people in general. Just people who are in an area that don't have a lot of infections being shipped to areas that do, so that they can run the equipment or help do the testing. There may be liability issues here in the US, but I feel like lawmakers would be able to make a law that allows this to happen. I would have liked to have seen that. The test kits were approved and were sitting on the shelf at IDT, which is one of the 2 places that makes them, for months before states started buying them. And I think it's generally been an uncoordinated response. You've got not even just individual states, but a lot of individual cities and counties that are kind of doing their own thing. They're all coordinating through states and feeding that data to the national level. But the type of response appears to be, to some extent, left up to individuals, and individuals vary. I'd like to see a more consistent response, and I'd like to see sort of faster, serious preventative measures and much wider testing. That's the stuff that would make me happy as a scientist sitting here with no public policy or public health experience. How long do I think it's going to last? I think it's going to last about maybe 2.5 months after we hit the peak, whenever that is. And the peak's going to be a little murky in the US because I think we're still eating into the cases that were already there and we didn't know about. So maybe 4 months if everything goes well, but everything is not going well at the moment. A lot of people are working super hard, particularly those medical professionals. And yeah, bless them and more power to them.
Okay, thank you. A note from Eric Schlauter again, who is the emergency management coordinator for Texarkana. Hydroxychloroquine and Zithromax are being used with mixed results for treatment, but obviously too soon to determine the true effectiveness, but worth continuing. Prevention certainly is the cure, maybe, and following CDC guidelines, practicing distancing, basically doing the sorts of things, kind of from a public standpoint, that would make sense. And again, kind of reiterating that this doesn't prevent it, but it gives a better chance to respond and prevent medical facilities from being overwhelmed, as well as to provide a change to develop treatments. So, okay. I saw one earlier about, here we go. Why is it that some viruses are seasonal and have previous SARS viruses followed this pattern? Things like cold and flu, we tend to think of a cold season, flu season. Is that real? Is there a reason? And do other SARS follow it?
Yes, no, yes. There we go. In a nutshell. So the 4 human coronaviruses that were in yellow at the bottom of that tree, those all do seem to have a season. I think they're not sure what the HKU1 season is. It just seems like it's baby season. Wherever there's babies, they get this thing. We are not exactly sure why some of these coronaviruses have a season. Coronavirus season tends to be winter to spring, so it's a little later than the typical flu season. The only one that we really know for sure is the Middle East Respiratory Syndrome coronavirus. That one comes to people. The bats poop, and the camels pick that up as they're eating grass, and they pass it on from mama camel to baby camel. And when the babies start to get infected, that is when they start infecting people. And so every spring is when the baby camels are born, and that's when you get a big old peak of Middle East Respiratory Syndrome virus. There's going to be reasons behind all these other outbreaks, but we don't know, and we're not even sure what the host is of that. What was the rest of that question?
That's fine. Basically, what accounts for the seasonability, and then
we don't know.
Is this a SARS thing? Like, does SARS do the same thing?
No, SARS did not, because we got rid of SARS, we hammered it, and it was gone. So SARS season was when it jumped from one bat to one civet to one person. And that is a weird chance event that's not going to be reproducible. That's not going to happen every year. And so, yeah, thankfully there is no season for SARS. And I'm hoping very much that there is not a season for the new virus.
Yeah, amen.
Yeah, I don't think we rely on spring or summer to save us. I see a lot of that nonsense in the news. We've got to socially distance. We've got to test. We've got to root this thing out on our own. There's no cavalry coming in. We got to do this ourselves. Yeah.
I would not necessarily prefer the truth all the time, Ben. Just saying. But,
sorry. All right.
I'm trying to see if I've missed any questions from earlier on. I think I asked all those. Here we go. I think, yeah, we might have talked about this a little bit earlier. Essentially, any information on the incubation period for COVID-19.
Yeah, so I haven't seen a good study where they could pin down the exact date where somebody was exposed yet, because there's still enough cases out there we don't know about. For SARS coronavirus, we do know exactly the date from some early stuff that happened in a hotel. And so it looks like the 2 to 10 day window is about right, with probably an average around 7 days. So about a week between when you're infected and you start to have symptoms, another week till they get really serious, and then you're over it pretty soon after that.
Okay.
So the virus lingers for 2 weeks, yeah.
Cheerful. All right, any last questions from the audience here? I'll give them a couple of seconds to type in very quickly if they have any that we want to talk about.
Thank you all for showing up for this weird thing.
Yeah.
By the
This is the new normal, man. We're
Sure.
all living virtually.
Webcams are amazing. Yeah. Huh.
Um, all right. Another from. Oh, here we go. One more question, then I'll sort of finish it off. If you've had pneumonia already, are you likely to have a more severe illness, or children with asthma? Is that going to be worse?
All those things seem to be complicating factors, but your age is the biggest determinant that we know of so far of how it's going to go. So if you're a kid with asthma, you'll have slightly more severe disease than any other kid, but you're still likely to bounce back really quickly, because those statistics are grouped by age. They're not grouped, the ones I showed you, they're not grouped by pre-existing condition. And so for all kids, yeah, the virus just bounces off them. Kids are Teflon, yeah, which is great.
Okay. And then one other question about masks. Are fabric masks helping? Is that basically to prevent you from touching your face again? Is that the idea?
Yeah, it'll stop you touching your face, and it'll show everybody else that you care. Yeah, so yeah. I think they're all right. Make your own. Yeah, it's a nice little quarantine craft project. Yeah.
Okay. All right, one other quick one. Sure. I just say it's quick, so take as little or as much time as you want.
Sure.
Why are some countries not seeing the high infectious rates that many of us are?
Well, if you look at the distribution of the coronavirus and you look at the distribution of international flights, the two overlap pretty much exactly. So as places with a lot of traffic are getting the virus first, I think eventually the virus may get everywhere because there are enough places doing a lousy job containing it that it is still spreading. And so the thing with a virus like this, when even one person has this, the entire world is at risk because we're all connected, which is why we like to see these things stamped out with great authority as soon as possible.
Okay, and there is one that I missed, so sorry. Someone has said, I've heard on the news that in some vaccine trials, not necessarily coronavirus, that the trial vaccines have made things worse. Do we know the biological mechanism behind that?
So there's a cat coronavirus vaccine that used to be legal to give to your cat. So there's a cat coronavirus that when it infects, it actually kills all the cats that get it. It's really bad, called feline infectious peritonitis virus. And they made a vaccine for it, and they tested it out, and it made really good antibodies, which is one way you can look at whether a vaccine is working. The thing was, the cat coronavirus is actually not very good at grabbing onto cat cells. It's really a dog coronavirus, or rather a hybrid between those two. And those dog coronaviruses need a little help. And so if you have antibodies, they'll actually act as a bridge. They'll stick to the cat coronavirus and stick to the cell it wants to infect. And then bam, now the infection is greater. And so you had this going from a disease that killed cats in 2 weeks to a disease that killed them in 5 days. Yeah. And so there are lots of viruses where we know something like this can happen. Things like dengue fever virus. You have to watch how the vaccine for that is administered. Yeah, so this is my concern when they're just putting in the spike protein, that they're going to get a great antibody response, no T cell response, and you could potentially make the disease worse, yeah, than it is. But we're not going to know that until the tests are done. And right now, it's great that the tests are being done. So,
okay. Do you know if it's true that people who are taking ACE inhibitors are less likely to be infected?
The ACE inhibitor binding pocket is different than where the spike binds. We know that when the spike of the virus binds, it causes the whole ACE protein complex to be pulled inside the cell, so it like disappears from the outside, which is why ACE doesn't work when SARS is there. But no, it seems like you can stick the inhibitors on there, and it didn't do anything in the original SARS. I very much doubt it's going to do anything in this one. They're binding to the same part of the protein.
Okay. All right, I think that may, one last one. Do you have any opinion on the use of the fabric masks by medical personnel working at the bedside? Do you think they're useful?
I want people who are working at bedside to have the N95s if they need them. There'll be some situations where it would be okay to have something else, but yeah, I would very much like for them to have whatever equipment lets them do their job in as greatest safety as possible, because those guys are the real heroes here. Yeah.
Okay. I think that's probably a very good place to stop, I would say. If folks think of any additional questions, feel free to, I won't barrage your email, but you can email us at honors@tamut.edu, and I'll forward them along to Dr. Ben, or you can email him if you want directly. That's fine.
Or go to our Dr. Ben, Ask Dr. Ben Facebook group, and I'll answer questions from anybody. Yeah.
This is true. If you want, Dr. Neuman has been doing a Ask Dr. Ben on Facebook. I think you're up to about 100 videos now.
Theme song. We got a theme song.
Got a theme song. It's you can go ahead and look for that group. It's is it just Ask Dr. Ben? Is that what it's called?
I think that's right. Yeah.
On Facebook. So you should certainly. I know you covered some of those questions here today, but there are many, many more that you've already gone through. And very entertaining and informative. So, all right, well, thank you. Usually we give you a round of applause, but I'll just give you a thumbs up for everybody. It was really, really good. And we'll do another of these in a couple of weeks. I'll put it on the Facebook page for, for the honors program. It's Dr. Ben Neuman's Science Group is apparently what it's called.
There you
go. And okay, thank you very much, Neuman. Appreciate it.
All right, thank you. Yeah, thank everybody. Yeah.